Yeah, it's here. It's in this room. Yeah, so for people that haven't been last week, let me make a brief summary of a few minutes about what we did, where I would like to go for the next 2 classes. And then the last class, so next week there is no class, and probably 2 more weeks. We will start, the final class will be about aggregation and fragmentation. So this class and the next class is going to be about polymer models for, so I'll give the introduction on this. Also how to use the polymer model when we want to interpret single particle trajectories. All right, so let me briefly summarize what we discussed last week. So, the idea is to look at modeling and asymptotics for basically targeted towards the, to asymptotics for nucleus organization. So this is basically the introduction. What are we interested in? Why do we need modeling? Why do we need asymptotics? What's the question? What's the motivation for what we want to do? So, as you know, a large class of cells, eukaryotic cells, have the DNA inside an envelope called the nucleus. So we have a cell, and here is the nucleus of the cell, and what you find inside is the genetic material. So what is the genetic material? So we have a precise idea now over the past 50 years that it is encoded inside the DNA, but the DNA is not like this. It is not this beautiful picture. The DNA looks like a bunch of spaghetti intermingled, and what is the sense of all of this? So over the past 10, 20 years, people wanted to find orders into this structure. So basically there are 2 sources of information. One coming from imaging. So this is the DNA inside the nucleus of the cell. This is a cell. This is a cytoplasm. And of course it is complicated. I mean, I'm not discussing here the fact that you have messenger RNA that can traffic. I'm just concentrating on these specific compartments. And the idea is that functions and structure are linked in the following sense. What is structure? This is this. So we want to find order into this. What is function is gene expression. What is meaning of gene expression? That is, a portion here of the DNA is going to be read by, when we have a transcription factor with a very specific protein that comes here, this is going to be read, and the output of this is to synthesize messenger RNA that will produce, then it has to translocate outside, and there is some machinery here that the messenger RNA goes here and produce a protein. For example, in the olfaction, you know, when we smell, how many different type of odors can we smell? About 800 different types of molecules. This is the code. Rats, about 1,000 something. Now why? This is still an interesting question and a mystery, that a single type of receptor is going to express in these olfactory cells. That is, a specific gene here is leading to a specific receptor, and there are about 800 of these in humans, in mammalians, I mean in humans. Once this one is expressed, the neighboring one, which should express a different one, is going to be suppressed. Not activation. How? Why? It's not clear. So there are all of this regulation people are interested in, of course. Right? So this is to describe that the structure, there's an organization here that underlies the function. Which one is going to be expressed? So I'm just summarizing what we did last week, you know, just about the nucleus organization. So we said it's a bunch of spaghetti. And now let's be more precise. So in yeast, in yeast, there is more order. That is, you have 32, so you have 16 chromosomes, 32 ends. The ends of the chromosome are called the telomeres. And here you see very nice organization where you have all the chromosomes that are pinned here at a point which is called the centromere. And then you see that, so the chromosome, they are making like an arm. Wait, I have to reattach the microphone. I'll put that in my pocket. Should be okay. And what people have found over the past 10, 15 years is that this end of the chromosome are exactly touching the envelope and they form cluster, which means that they come and they can spend time together. And what has been found is that you can have 3 to 5 or 10 or 8, maximum 8 clusters, and then you have a different amount of brightness, which suggests that the number of chromosomes or telomeres per cluster can vary. Now, why do you have this? This is an interesting question. So the idea today is that they can move and be exchanged in between those clusters. So this is to show that it's not just like spaghetti like this, at least for yeast. There is some organization. So now, if we go to mammalian cells, what has been found is that the organization is not exactly the same, that it seems that these chromosomes are occupying territories. What we mean by territories, it means that if you look under the microscope, this region, obviously what happened here doesn't mix with what happened in this region. So this is the idea that the chromosomes, by a mechanism that remains unclear, but can be understood by polymer simulations, that the chromosome will exclude the neighbors ones, and you have these nice regions. Again, there is some organization that we're interested in to understand how this is organized, which is different from yeast. Now, there is, so what are we interested in here? Another question is, for example, what happened after a double-strand DNA break? Again, I'm giving the different motivation for introducing the modeling and what we're going to do with these models. So suppose you have a cell. This is the nucleus. This is the DNA, or I call DNA, but this is really the chromatin. The chromatin is like all the molecules that are around the DNA that form this dense object that you can see under the microscope. It's more than just the DNA. You have what is called nucleosomes. You have all kind of very rich environment here, and it can be heterogeneous. Now, what happened during a break? There's two types of break: single-stranded and double-stranded. Double-stranded, you take a scissor and you cut literally. So you do something like this. This is quite important because this event, for example, happened when you go this summer in the beach and you just lie under the sun, and you have all of these events that can happen. And you know that it can lead to cancer. And this is exactly, for example, what radiotherapy is about. Is about not killing the cell by shooting the beam. It's you shoot the beam, it creates breaks, and if the cell is not able to repair, so you think that there are going to be a difference between cancer cells and normal cells. So if the cell cannot repair, it kills itself. Trigger apoptosis. Which means that, so how long it takes usually when you have a break? It takes a couple of hours. This is what people have found experimentally for events to be detected, that the cell is stopping everything, trying to see how it can repair this. And so this is still under investigation, how this is done. So that's why, for example, experimentalists, people interested in this, they can here put a label. That is, put something that will emit light or fluorescent dye, And you can get trajectories of this. That is, under the microscope, this is what, if you have a trajectory, so let me draw it in a different color. Now you have, this is not a polymer that I'm drawing. This is a trajectory. This is a single particle trajectory of a die here. Let's call it the point x of x. So you have x of t and t for a certain time window from t1 to t end. And of course, you would like to extract information from these trajectories. And now we would like to use the fact that it comes from a polymer to see what kind of information can be extracted. Right? So if you have this, can you extract information about diffusion, coefficient? Can you extract, for example, if you see a spot that is very confined, you may want to say that there will be a physical reason why it is confined. For example, you have a force that will bring this point at the center. But we don't know where the force is applied. Maybe a force is applied here. So a question that I would like to discuss today is, suppose you apply a force here, where suppose this is classical parabolic force applied at one point, and you observe a trajectory, a particle here. Can you, from this trajectory, or some statistics of the trajectory, recover some information about the distance from that point to where the force is applied? And can you get some information about the force itself? Right, so that's the ideas. Okay, so just to continue on this, so there are two modes of repair. Either you glue back this part with that part. This is called non-homologous end-joining. It happened in some conditions. But of course, if here when the DNA has been cut, some information has been lost, somehow the DNA has to go find out the sister chromatin, where is the information that is missing. So there is a search process, a blind search process. That is, you have to search for something that has some information for you, and you don't know where it is. It's like a random search, except that you are now attached, or you're bound to the entire polymer, and you're trying to find something in this nuclear space, and you have to get here, and in here, by a process that I'm not describing, you can copy back the information here, like what you do on the computer when you copy. You copy back the information, then it can be glued. Now, some drastic event can happen. Suppose now you have two breaks. Now, it can happen that under some conditions, instead of this being repaired directly by this one, this is going to be repaired by that one. And this one is going to be repaired by this, which is very dramatic for the cell. For example, when this happens, so you can see on the microscope, for the cell, it has repaired. That is, there's some machinery that tells the cells this has been repaired, and the cell can continue. For example, if the cell wanted to divide as a next stage, then because of this, you can see under the microscope that the cell tried to divide, but the chromosome cannot separate anymore because of this wrong repair, and the cell is stuck. That's why most of the time the cell triggers apoptosis, not to engage into this impossible division. All right, so this is the motivation of what we want to discuss. So let me continue with 4 statistical quantity that we would like to extract from the trajectories. And then we're going to construct the polymer models, or to tell you the polymer model that we need, and to investigate their property to understand how we are going to connect with the data. So basically, when you have empirical trajectory, I would like to give 3 statistical quantities, so 4 statistical quantity to calculate. So from single particle trajectories, we're going to get x of t for t in a certain interval. Usually it is sampled, it's a bit more complicated than this. It's not continuous, but usually you have x1, xn. And usually they are not necessarily representing the physical trajectory because this can be corrupted by some localization noise, for example. That is, you have dots, and then you have to attribute that these dots are part of a trajectory, and you can make error during this process. So then, suppose you have this information, can we calculate? So we are interested in calculating, first of all, can we from this estimate the diffusion coefficient, which gives some information about the second order statistics of this. We can, so if we have a region where it is confined, basically you are interested in evaluating or estimating from a single realization information about the confinement. So here it's clear that if you have such a shape, you can approximate this as using a disk. In general, it's more complicated when we have something that has not such simple geometrical picture.
Excuse me, in your notation, x is a vector that locates the particle in space?
That's right. X of t can be, for example, x of t is, for example, if you have x1, x2, xe, you just take a reference, you take a frame, x and 1, xe are the projection, so you have x, y, and z. You have this, so x of t, this is what I meant.
Okay.
Diffusion coefficient d, then so you're interested in the radius of confinement. So then there is what is called the mean square displacement, which everybody has calculated, which is you take x of t, let's suppose at 0 it is located at 0, you average over realization, and for short time asymptotics, it behaves like a constant t to some power alpha. So usually it's of interest to estimate alpha. Estimate alpha and a. When alpha is different than 1, you have the three cases. It's larger than 1, bigger than 1. This is related to anomalous diffusion or subdiffusion or superdiffusion. Still, to that day, it's not clear what is the meaning of alpha. So is it in the case of a polymer moving, is this giving information about the internal property of the polymer? Or is it giving information about the statistics of the environment? So what?
How does that differ from the second one, the diffusion coefficient?
I'm sorry?
Isn't that the same as the diffusion coefficient?
Well, the diffusion coefficient is when alpha equals 1, you have this A, which is connected to D.
Yeah.
When alpha is not 1, then this is something else. Because you want, I mean, this is assuming, that's right, this is assuming, this is also second order property, you're correct. I should have probably inverted the two. This is for alpha equals 1. We are in classical diffusion. You extract what is the diffusion coefficient, which is basically contained in the A here, depending, and there is an additional factor of the dimension. So the question is, what is the interpretation? What is the interpretation of alpha? And the last quantity of interest is the first order property, the first moment, really. And so the first moment is associated to the force. Can you, if you have a confinement object, is this confinement due to a force that pull back the trajectory? There's a force. So for this, we need a model that I will give in a second about the dynamics, such as the Langevin equation or the Smolokhovsky limit. So then, can we extract the force or the mean force applied to x. So this is 1, 2, 3, and 4. All right, so this is related to what is called single particle trajectory. That is, you want to extract information from dynamical data. So again, why we want to do this? For example, we want to look at the situation before a break, after a break. Are those quantity changing? Can we classify the different, what happened depending when you are, if you have a spot that is different located on the DNA, can you see differences in those 4 quantity in the statistics?
So David, this last one, this mean force, that's applied at an unknown position.
Yeah, So this is like a tethering force. It's like, think about it, you have a potential well somewhere, and you have one of the beads of a polymer. So these are beads. They're connected by a spring. And suppose, this is now we're entering already into a model, into a certain vision of what we meant by force. Suppose this bead, which is part of the polymer, simple Rouse model where you have a spring constant here, and this is, you have here force which is coming from this potential. Like u of x here is like alpha x, so alpha is a constant. Now suppose you take the gradient of this force, so if you're in dimension 2, this is x1 squared plus y squared, and you take the force, which is a force that's prevent basically this guy to move. I mean, you move in this bottom of the well. This is what I meant by a force. Now if you put a label on your polymer, let's say here, this guy will feel some of this constraint. Now can you recover this information here by looking at this this guy, or even by just looking at this guy here? You look at this monomer, you look at this trajectory. Can you reconstruct the force? There are different reasons to be confined. You can be confined because you are confined by obstacles. But this gives different statistics. I mean, it's also important to discuss that it depends, of course, on the sampling rate. You have to sample, you know, if between 2 sampling you have time to explore. It's not the same that sampling at a much faster frequency where you get information really about the underlying physical process. Okay, I'm not discussing this right now, but all right, this is the notion of a force that we have in mind for this. So there is one more thing that I would like to discuss for motivation, so we're still part in the introduction, is the new type of data, which is called IC data. So IC, or chromosomal capture data. So suppose, let me describe very vaguely what are those data. Suppose under some condition you can freeze, literally, the DNA. And so you have many copies of the cell. And you're looking, so you suppose you have 1,000 or 1 million copy of this. So this is a different realization. And then suppose you are able to look at the frequency in how many cells. For example, do you have, let's say, point or the genomic, suppose you can measure exactly where you are in these genomic distances. You have I and you have J here. And you look, what is the fraction of time the two are together? Together means, let's say, fix a certain distance, epsilon, and you look, what is the fraction of time they're together? Now suppose you can do that for all I and J. So then you have what is called, you are going to get, by averaging or taking all your ensemble, a frequency encountering matrix. What is the fraction of time the two are together? And so it turned out that there are specific, so this leads to what is called the encounter frequency matrix. And so basically, if you have, depending on the scale at which you are looking at this process, you can end up having matrix of 10,000 or 1 million by 1 million, like very big matrices. And events such that suppose you have, each time you have the same A and I and J, suppose you find that they are more or less together. That is really, they are, for some reason, forming a loop. Yes?
Just a couple of questions. So how do they do the microscopy? So they have like...
Well, the microscopy, this is, it's not under this is, so you do formaldehyde, and then there is an entire process of you kill the cell, you do the digestion, and then you come back to this picture. It's not under the microscope that they're seeing this.
Is it done through fax machine somehow?
So what? I'm sorry?
Through a fax machine or something like that?
I don't know what that is, fax.
Well, flow cytometry.
No, I don't know. I don't think so. I think there is this is a complicated process that has to do with genetics, with looking at the different, recognizing the different base pairs. And it's not under the microscope that you see that the first part, which is just single particle trajectory on live microscopy. This is a different technique.
Is this FRET?
No, it's not FRET.
Okay, so how are they, that means they have to take a photograph somehow.
Or sequence. Or do they sequence this?
They looked at the different, so for example, they go and then they have to look at all of these different segments, the information when they glue together, then they cut everything, and then they see which guy is with the other guy.
Oh, okay.
It has nothing to do with
so this is actually the distances in R. The distance is not necessarily physical distance.
That's right. No, I'm saying they're together. Okay. But then you know where it is. Then you have to go back to the bioinformatics. You know where the sequence is. You know where the sequence is, and then you can whatever happened in between. You don't know, but they are together. This is partial information, not and so what you're raising is the important question in the field, is that suppose now you get this matrix. I didn't draw this, so you have this lambda ij, which is your frequency of encounter. Can you? This is exactly the question. Can you reconstruct the 3D geometrical organization? This is nobody knows. All right, but let's write the question: how to reconstruct the three-dimensional chromatin organization from the, let's call it the encounter frequency or the encounter probability matrix. So in particular, there is a question that we would like to discuss, which is a theoretical question. If the method is sensitive, basically, or if you get all of these events where two sites which are located far away are not together. And this happened many times. It means this loop is quite important. Now, you can ask the following question: is it a purely rare event that you form a loop and this is what you get, or there is more? So obviously there is more, but the question that we would like to answer here is, suppose we have no connectors. That is, you can say that you can have a loop because you have indeed molecule, and there are names for this molecule such as cohesin, that comes and attach the two together. And there is probably a good reason for this. But suppose now you don't have this, and now you have a polymer, like a Rouse polymer. So how long does it take, depending on the length, to form a loop for the first time? which will give us the rate of mean looping that you can get in this ensemble depending on the length of the polymer. So the question is, what is the rate of looping? Or 1 divided by the rate is the time. So what is the mean first time for a loop to be generated?
Question. Why do I care about the rate? I don't get to watch this dynamically. I only get to see the steady state.
That's true, but the mean first passage time, it's one over the rate at which it happens. And now you can get the information about this matrix by normalizing this rate. So for example, if you are interested in the probability that you have A and B here together. So what is the probability that you form a loop TAB before any other loop? So then you can calculate the mean first passage time for this, and you normalize by the sum of the mean first passage time to the other, and then you get steady state properties.
Then you have to use some dynamical model that you're not actually.
Yeah, I haven't entered into dynamics yet. I'm just putting the questions.
Sorry, the loops can't break.
Sure.
Yeah.
Okay, so in a second, I'll give you the model. We'll start with the model, and actually what I would like to show is that now we can calculate the frequency of forming loops. And again, this you can see it as steady state once we have the time of formation. So now, in case of a Rouse polymer, so I'll explain what we discussed a little bit last week. It's the Rouse polymer and its generalization. So the formula of interest, so the result I want to discuss today, is that the mean first passage time to form a loop, let's say, between the two ends of a polymer, of, let's say, you have the number of beads is there are 1 to n beads. This is the total number of beads. So the mean first passage time, which is the average of the time, is actually something that you can calculate asymptotically for n large, and a given epsilon. Epsilon is the, let's say, you fix a radius of size epsilon, and you set form a loop for the first time when the second extremity, this one, is going to enter for the first time in this ball of radius epsilon. So the mean first passage time turned out to be, so this is what I want to discuss today, n pi divided by kappa. So I'll explain in a second the notation, and I will introduce the dynamical model. To the power 3/2 square root of 2, d diffusion coefficient 4 pi epsilon plus a constant that can be calculated, A3, b squared, this is the mean square displacement between two neighboring beads, n squared plus O of 1. And the expansion is for n fixed and small epsilon. There are two parameters here that are important. So n is the total number of beads, and epsilon is the radius of the ball of encounter. This is in dimension 3. So A3 is a constant, D the diffusion coefficient. All right, so to explain what is kappa and B, I need to say that this is calculated for what is called a Rouse polymer. So now, maybe this is really part 1. So this is an asymptotic formula that explains how the looping time depends on n to the power 3/2. Kappa is like the spring constant. Yeah, and B is the mean square displacement between two neighboring beads that you have here. You take this mean square displacement here. So we know exactly how it depends on the total length, n, and epsilon. So this is a rare event. You have to wait. We're going to see that you have to wait a long time compared to, let's say, any motion here of the internal beat for this event to happen. This is reminiscent of a search time of diffusion to a small target of radius epsilon, except that now it doesn't depend. I mean, usually you have here the space. It's the space, omega, the volume, divided by epsilon. Here this is something else. This is the total number of bits to a certain power, and it depends on this coefficient kappa. And then there is an additional term which does not depend on epsilon. This term has to do with the internal property of relaxation of the polymer. People have discovered this term about 30 years ago, because when you have a long polymer, you stretch it, you let it relax, and if you ask the same question how long it takes to find something of size epsilon, then it becomes the dominant term. It was independent of epsilon. This is how they figure out that there was this term. Under some other regime where epsilon is very, very small, they could not see this term. Then they had this term. This was the Pastor, Jabot, and another fellow that discovered the first part of the formula. So actually, you have the sum of the two. So this is dimension 3. You can also do it in dimension 2, which I'm not going to write it here, but to have just looping in dimension 2, which is not necessarily relevant for what happened in the nucleus. All right, so let's...
Is this chain completely flexible in some sense? That
yes.
Each
yes.
node you can...
Yes, I'll give the model now. So let's go to part 1 for the model. And I want to discuss this Ross model because, so last week, this is where we were last week. We started from the Ross model. I mean, this is an introduction, why we want to do this, what kind of question we want to ask, and where we are going, what kind of calculation we want to do, especially this falls into a new type of narrow escape problem. How we found this. The idea here, compared to classical narrow escape is the mean. So let me just make a comment. So the classical narrow escape problem is what? You have, which we discussed a couple of class ago, you have a random particle, and you have a small hole somewhere. And then you ask how long it takes to find the small hole. When you have a diffusion, now let's discuss dynamics. You have x dot is dx dt, equal square root of 2d, the diffusion coefficient, and w dot, the white noise. The mean first passage time to this, we say it's d Laplacian u, equal -1, u equals 0 on the absorbing boundary, d omega a, and du dn, the particle is reflected anywhere else. It's on the reflecting part. So this is a classical boundary value problem, which is solving the Laplacian. And the asymptotics, either using Green's function or match asymptotics, you find that u epsilon of x is omega, the volume. So let's say in dimension 3, divided by 4dA, plus there is a term here, which is, I forgot exactly what it was, but it's usually the mean curvature at the point here, if you have a regular point, divided by pi or 2 pi, log of 1 over a, plus the term of O of 1. And in dimension 2, this is omega log of 1 over epsilon, where epsilon is the ratio of the absorbing boundary divided by the total boundary. And this is much smaller than 1. And a here, this is the radius of the absorbing patch. Suppose it is circular. So a here is the radius of the absorbing window, which is circular. All right, so this is not exactly falling into the same scenario where you just have to solve in dimension 2 or 3 the Laplace operator, because the problem is it's not one particle. All particles here are correlated. When this one is moving, this one is moving, this one. So to solve this problem, we didn't know how to do the same. So you had to go really to the high dimensional problem where you look at all beads. So the dimension is the space for this multiplied by the space for this, because you have to consider the joint probability distribution function. I will explain this. So that's why here you are in high dimension. And you have to solve, basically, a problem of finding a small hole for, let's say this one is fixed. You can always pin the polymer. Is the translation motion is unimportant. What you're interested in here is what happened as a rotation event. So the translation is unimportant. When this guy is entering into this region. And so we will see that this is like the boundary of a submanifold in very high dimension. The dimension is n, the number of beads, multiplied by, if you are in dimension 3 for each bead, so it's 3n. Right? So this is what I want. The comment is that the looping time, the mean first looping time is resolved, or has to be resolved, is resolved in the dimension r, let's say 3 to the power n, if you're in dimension 3. So you're in very high dimension. If n is the number of beads, for example, if you have a polymer of 100, 200, and 300 beads, so n is the number of beads. So that's why I made this very brief remark. So you fix n, and then epsilon is small. But what you are interested in practice is, for example, for very long polymer, n being very large. The problem is this formula is incorrect if you exchange the limits. So this is important to notice, because this is what we were interested in looking for, a sort of uniform asymptotic formula. All right, so I want to discuss this. So now let's discuss the polymer model. So this is part 1. Let's discuss the polymer model for this and the associated data polymer that we have developed to answer the following question. What is the property of a polymer that has a fixed anomalous exponent? Suppose alpha is fixed. So for a Ross polymer, alpha is equal to 1/2. This is known. Now suppose you do a measurement. You take alpha. It's coming from the measurement. So remember, we ask, what can we say about alpha? What is the interpretation of alpha? So what we would like to claim here is that alpha can reflect information about the polymer itself. Just we have to understand how the different beads are connected. They are not going to be any more connected through the neighbors, but they can have long-range connections. And the question is, how do you prescribe those long-range connections such that when you calculate the anomalous exponent of this guy, it gives us alpha? All right, so let's start with part 1.
I'll just ask you, just on your comments. So if you were to do just diffusion in 4 dimensions, would the mean first passage time scale like 1 over epsilon squared, or still just like 1 over epsilon?
No, it could be like squared.
Okay, so then I'm curious why in the high dimensional space you still have the 1 over epsilon scale.
This is a very good question. Because here what is it's the dimension is not important. What is important is the codimension. Because you don't have to look. Imagine that in dimension 2, you're not looking anymore at a small hole, but you look, let's say, at a cylinder. And now it's different. You look at a 2-dimensional object. You see what I mean? So this is what happens. When you increase the dimension in here, you increase also the dimension of the subregion that you want to find. And the difference of those 2 dimensions, this is what it remains 3. Because you're going to have 3 constraints. That's the reason. It's clear, right? So I'll come to that. Okay, so let's start I'm looking for.
Behind your
Yes. Okay.
or behind your sweater.
Yeah, I put them all over the place. All right. I have to. All right, let me erase this, and now let's start with your question about dynamics. What is the dynamics? How we see this? What is the polymer model? So this is part one. This is polymer dynamics. So all what I'm discussing here is based on classical polymer models, but the analysis is the new part. How we analyze them compared to the steady state analysis is present in all very good classical books, like Doy-Edwards. I mean, there are infinite number of very beautiful books about this. Doy-Edwards, there is about polymer model. I think it's 80 something, 84 or 85. There's a book of Rubinstein about polymer model, and there are many very interesting books. So let's start with the Ross model. The Ross model is beads connected by spring. So you have an energy. So let's call R1 Rn the position of the bead. It can be time dependent. We're in dimension 3, so this is a vector or point in dimension 3. And so now we need an equation for equation of motion. So we took the Smoluchowski, I mean the classical version is the Smoluchowski limit of the Langevin equation. So you have just a motion, an equation for the derivative of the position, which is the velocity, which is minus the gradient with respect to Rk of the potential, which I'm going to write in a second, plus noise, square root of 2d. Let's suppose d is, you can attribute whatever you want. So let's suppose d is a diffusion constant for this bead, which is constant along your polymer, plus your Brownian noise. And the U of R, which is the energy, is U of R1 Rn equals kappa divided by 2. So this is the same kappa that we've used over there. This is just a spring constant. Sum of Rk minus Rk minus 1 squared, and k has to go, let's say, from 1 or 2 to n. All right, so for example, if you have if you look at the monomer, let's ignore the two end ones because this guy, they don't have any more a guy here to connect. But if you're in the middle, basically you are connected to the two neighboring monomer, and the force is simply the spring constant that's bring back the monomer to a position. So for example here, general equation is for monomer that are inside. This is 2Rk minus Rk minus 1 minus Rk plus 1 kappa plus noise. And so, and for k from, let's say, 3 to n minus 1, the extreme part, or 2. The extreme part are different. One term is out. And so last week, Kevin made a comment that this is really like a Laplacian operator, discretization of the Laplace operator in that one dimension. And this is important for understanding what can be done with this polymer to create a polymer a little bit more complicated. So this is Ross. It has interesting properties. You can look at okay, let me remind you some of the basic property, and then from here, so basically what we're going to do is you can find a base that will diagonalize this matrix. And we'll see it makes sense to diagonalize. And with this diagonalization, we're going to construct a polymer. Will have a predicted, a fixed alpha exponent. Okay, so some properties. Again, this is very classical, but so Rn in steady state minus the position, R1 minus Rn, for long time asymptotic, so you can look at it at infinity, is basically the mean, when you average over realization, it's of the order of n b squared. So I haven't said this, but the mean position, so b here, it was b is the mean position between R1, Rk, any of them, Rk minus 1. Suppose this is fixed. This is the second order properties. All right, and also an important quantity to characterize the polymer is called the radius of gyration. Radius of gyration, basically, we have a polymer, and by symmetry, you would like to ask what is the radius where this polymer is confined, and the radius of this is called the radius of gyration, Rg. And Rg for a polymer, so it's defined as you take the sum of the position Rk, you take the mean position, let's call R mean. R mean is the average over R1, Rn. You take its square, you take the ensemble of this, and this behaves basically as square root of n. And there is a constant here, and the constant, so it's b divided by square root 6.
Is b the square root of what you wrote up?
No, b is...
What you've defined as b?
b is here. Yeah, b is square root of this, sorry. Okay, so now let's go into the study of this. Not that it's technically difficult, but this is the frame we need to construct the other polymer. So let's diagonalize this. So I'll give you basically the matrix that we need. So let's define UP, which is the sum of alpha Pn, and with sum over n from 1, I think, to n or n minus 1. It's not written in my note. So alpha P1 multiplied by Rn. Yeah, you sum over all of these. And let's take alpha Pn, which is the matrix, which is 1 divided by square root of n, if p different if equals 0. Otherwise, we'll take 2n square root cosine n minus 1/2 p2 divided, so 2p divided by capital N. So using this transformation, you can write an equation for UP. So you differentiate this with respect to time, du dt. So this is independent of time. So this is the Rn dt. And drn dt, this is given by this. So I'll skip some steps. Basically, this diagonalizing this, so you are going to end up here with a single term. And I'm going to write an ensemble of Orstein-Uhlenbeck processes, which is du dt of UP. It's minus d kappa p up plus square root of 2d w dot tilde. So let me explain. This is the Orstein-Uhlenbeck process. D is the diffusion coefficient. Kappa p, this is the interesting quantity, which is 4 kappa. Kappa is a spring constant defined here. So 4k sine p pi divided by 2n squared.
These are the eigenvalues of that.
Yes, this is the eigenvalues of this. So you have this diagonalization. In here, what usually you have is, because of the sum, you are going to sum noise. But if you sum all of this noise, of these noises, this is like a single Brownian noise, and this you can see that it doesn't affect, the coefficient is exactly 1. So you just call this as a new noise, and this is what you get. So this is much simpler to study. From this you can calculate immediately, for example, the mean, the second order moment of UP, then plug, so you have this matrix basically is orthogonal, so you can basically Rn is the same alpha pn as a function of UP, so you can get all the property you want. So this is important when we are going to calculate the second order properties. And so let me so this is true for p equals, it's not written here, but 1 to n minus 1. There is u0, because we have n beads, so from 1 to n. This is n minus 1, but u0 is like the motion of the center of mass. That is, you have your entire polymer that moves. So u0 is just the motion. U0 is just the motion of the center of mass. That's why, if we're interested in looping, for example, we don't care about u0. All right, so maybe I'll go.
What you've done is you've diagonalized the
Yeah.
into a set of Orstein-Uhlenbeck.
That's right. That's right. I mean, this is the classical. I haven't done anything right now. This is classical. Now, let's discuss. Okay, let me introduce at this moment. I'll come back to here later. Let's discuss what you can do with this. Suppose instead of 2, I call it beta. I change 2 by beta. If you go back, so this is a reversible process. If I have 2, you can go back in here. And now what happened, so in the Fourier space, you do your transformation, you change a coefficient, and now you want to see what happened when you go back. You have modified here, basically, your eigenfunction. Now, before this, the eigenvalue. Before, the eigenvalue was such that everything was cancelling, such that you had only interactions between the neighbors. Now, because if you change the beta here, I mean, you change 2 by beta, you want to see how it propagates. All right, let's do this. And then let's calculate for when we are going to change here, let's calculate the mean square displacement of one of the monomer and compare the situation when we have 2 and when we have beta. And now, what I would like to show, I'll write this and then we'll make a 5 or 10 minutes break. We are going to see how the alpha, anomalous exponent, are going to depend on this beta. So right, this is where we were last week, more or less. So let's do this.
So couldn't you do this quickly with spectral calculus?
I don't know, maybe, what do you have in mind?
I mean, essentially, you just are taking all the eigenvalues and you're raising them to the beta halves power, right? Then presumably that means you've just taken the operator in your equations and changed it to the beta halves power of that. and then go from there.
Okay, but what I'm going to show you here is that now we have a procedure of calculating, implemented it in a computer manner, I mean calculable, when you want to calculate things, that we are going to be able to have explicitly, because we work in this Fourier space, the long-range connections. So we cannot do the calculation. We cannot run a polymer, a beta polymer. We make this long-range connection, and it will have alpha as anomalous exponent. So we have an algorithm procedure. Can I do that with what you said?
No, I think this was related to what Kevin was talking about last week, where you can just think about.
You could do it, but I'm not sure. I think this would be more insightful. But you could do it. Yeah.
This is a construction procedure.
Do a spectrum.
Sure.
So I mean, if while I'm writing all of this, you have an idea to simplify it, this is very interesting. I mean,
It's nothing more than what Kevin said last week. So.
okay, all right. Okay, so now the idea of doing this is now it's a constructive procedure. All right, so let's so the potential, the U, I'll just erase it. Okay, so let's call it U R1, or the potential, which was R1 or Rn. It can be also written as the potential of U1 Un minus 1, and this is exactly one half of K tilde P. So K tilde P is Kp, where I change 2 by beta. Up squared. So this before, let me remind you what it was. It was a 1/2 kappa sum of Rk minus Rk minus 1. So here I'm skipping one step. The step is to write or change Rk by the expression in terms of, so yeah, maybe I should write this down. So you can invert the equation. So let me write this somewhere. C is any of the monomer. This is P equals 0 to n minus 1, alpha Cp Up. So this is basically, we wrote U as a function of R, and this is now R as a function of U. This is just to invert it. So if we replace this in this formula, you get this sum of squared here. All right, so now let's go back. Suppose, so this we did with factor 2. Now you take the same formula, and now you put here the kappa p, which is sine to power beta of what is written there, which is 4 kappa pi p divided by n of 2n. All right. So when we have squared, everything is going to be beautifully like this. Many cancellations. Now, in general, what you get from here is we get 1/2 of the sum of kappa p, and then we just replace by this formula, which is the sum of alpha pn, and this is what I meant by explicit way to calculate the different forces. So this is squared, and now you can expand this sum. It's nothing very creative, and you reorganize it, and you can always write it as the sum of RL, Rm, ALM. And now for this ALM, this is the matrix, which is the sum of 1 over n minus 1, kappa tilde p, alpha PL, alpha MP. So just to show you what you get 4 kappa. There is 2, so this is 8 divided by n, sum from 1 to n minus 1 of sine beta p pi divided by 2n, cosine of p minus 1/2 p pi divided by n, and it has to be symmetric, so I have to multiply here. It's not great, right? So I multiply here, this is multiplication, by the same term. Sorry, this is sum over L, so there is an L here, and I have to have an M here. So this is cosine of the same term, so this is M minus 1/2 of p pi divided by n. So believe me, when you have squared, you have all cancellations. When you have a general term, you have no cancellation. So this will give you the information, so you can always rewrite this potential, so let's call it psi, as 1/2 of the sum of Aij Ri Rj, which is now the connections. This is A here is the connection between any monomers. You fix I and you have all the j's. So now you can expand this so that it is equal to this, and you get the information about your Aij, from which you can now run a simulation. A simulation. Now let's break here. After the break, we'll show the property, which is the interesting property, is that, so after the break, I would like to show, so that I make sure that you're coming back. Yeah, I think I lost it. Okay, I have to. So what I want to show after the break is that if you take not the end one, but telomeres in the middle, monomers in the middle, the mean square displacement, so T0 is fixed, let's take 0, take this squared. When T is small, these behave like a constant, and T to the power, I think it's 1 minus 1, minus 1 divided by beta. For T small, A is a constant, and the beta here is the same. So we're interested in when this quantity is positive and it's going to be less than 1. Basically, what this says, the conclusion I'm giving the conclusion before we have proved this. The reason, I mean, it's not only reason, a possibility, or when you extract an anomalous exponent from a tagged monomer, this says that it is contained. You can have, let's say, between 0 and 0.5. If you have a prescribed alpha, that is, if you look at your measurement and you get 0.4, for example. All right, you invert this formula, you find beta, you can do the construction, you get this long range. So I haven't shown to you, but the long range connections are decaying very quickly. Basically, you connect the two, but all of them have to be connected, but that's very small. But it's very important. You need this connection, even if it is small, so that this is prescribed. And so the one possible interpretation, I mean, is that the anomalous exponent comes from the properties of the chromatin. It has nothing to do with the local interaction, which is a question that is under debate. What is the why you have an anomalous exponent? And there is more. For example, for the same chromatin, depending where you are, the same chromatin, the same point that is labeled, you find different alpha. Why? A possible interpretation is that the chromatin contains different type of molecule, different type of binders. But at least from the analysis of the polymer, we can say we are able to construct a polymer, given polymer, with a prescribed exponent. Right? So I'll show that when we come back. And also, I want to do this, and I want to do the mean looping time, how long it takes for a Rouse polymer. So we did also the looping for a beta polymer. That is, what happens when you have all this connection, you take the endpoint, how this will affect the mean time it takes to loop. And actually, it affects, because instead of having the power 3/2, which was written before, it's now a power that depends on beta. All right, so I want to do this, and how long it takes for when we have a Rouse polymer for the two endpoints to meet and to recover the formula that we had on the board, and what is the method? Because the method is very different from all the methods we have developed, either us or Michael Ward of the group at University of British Columbia. It's not based on match asymptotics, Green's function. It involves a theorem that was developed about 20, 30 years ago. When you have a space and you remove an ensemble, what is the time it takes for a Brownian motion to remove an ensemble in very high dimensional space? And basically people looked at this by saying, what is the shift in the spectrum of the Laplacian? That is, you have a domain, you have some boundary conditions, you can calculate the spectrum of the Laplacian. Now suppose you remove a small domain. What is the consequence on the spectrum? So this is what they calculated. It has a shift in the spectrum, and the shift is exactly what you can use to connect with mean first passage time. So this was a completely different approach, and so I want to discuss this for the next hour, and if we don't have time to finish, I'll discuss this also for next class.
Very good. I thought Word and Color did precisely take a point of view of, you do some localized change, cut out part of their domain and look at the change in the spectrum, and that's how they got the mean first passage time results.
Oh, who did that?
Word and Color, no?
Michael. Well, how they did it? How they did it? It's a good question. The 1993 paper in Science Journal of Applied Math, this is probably what you have in mind.
All I'm asking is, it sounded like you were saying something is different.
It's different, yes, it's different. They did in dimension 2, dimension 3, by a process which is match asymptotic. Okay. That is, you look at a solution near the target and you glue far away. Oh, here it's going, for what we did for the polymer, it comes from a different vision where you have to remove something that has a structure. That is, it can be torus, it can be, it's basically the tubular neighborhood of n-2 or n-3 dimensional manifold that you are removing. This is very different. And their proof, one is probability proof, and the proof for Michael is gluing. You rescale everything in here, you find what is called an inner layer, that is some kind of how the solution behaves. And this is basically pushing everything at infinity, and then you have to connect with what's called an outer solution. And this is, you need for this what is called the solution of the Weber electrified disk problem, which is well known. You have an electrified disk, and you look at the potential far away. This is how this method is constructed. Nothing to do with this one. Okay, let's break, let's say 10 minutes. That's okay? 10 minutes, and then we will see how good, maybe I'll do only a half of what I said, and then I'll postpone it for next week, depending on how you feel. Okay, next week, no class. I will say it because once it happened that I forgot to say it, but next week, no class. And then the week after, we'll resume. Next week, no class.
That time some guy from Schlumberger showed up, right?
I'm sorry?
And that week someone from Schlumberger showed up to sit in on your class, right?
I think, yeah, somebody showed up and he was very upset. So I wanted to make sure that, and then the director came, and, you know, you have to be there when you give a class, but I said it, I said that, I didn't say it for the announcement. All right? Okay, let's break 10 minutes. All right. Okay, so we should start now, either by doing the looping or doing the construction of the beta polymer, of the mean square displacement.
Did you discuss whether the Rouse model is a good model of DNA to begin with?
I just got this question.
Yeah.
Yeah, so there's a debate, and there was a couple of publications showing from an experimental point of view that the alpha was quite good. The approximation of alpha is 0.4, 0.5,
This is
0.5.
for the anomalous diffusion.
Anomalous diffusion. So it came from this literature. The argument came from single particle trajectories and from the alpha. That it was quite a good description, although there are
So the alpha you get from experiments is close to the alpha you get
the mean is close to 0.5, although for bacteria, the mean is 0.4. And there is a large variability. That's why we built all of this polymer model to try to account for this spectrum. So this is how we saw this construction of the polymer to go in that direction.
Well, I asked him this earlier, he gave a better answer. It's because it's on a big scale. All of these twist bend coupling and whatever worm-like chain is apparently small scale, and he's on a big scale.
Larger scale like nucleosomes.
Right, but I mean, if you want to tie this down to actual distances in space and things like that, you have to say what D is and
Sure. Right now it's just about
qualitative features of the.
that's right, how things behave. For the connections, so now, I mean, the important question that you're raising is, now, what is the scale we are discussing?
Yeah, what's
here?
the scale?
Yeah, is it 1 megabase pair? I wanted to postpone this later. So actually, you can decide, you can choose. And so, for example, recently we looked at this chromosomal capture data, and one bead was representing 3 K base pairs. And at this scale, this is a reasonable assumption. If you go smaller, you can go to all kind of problems, because the chromatin is not just like presented like this. It's much more complicated. You have the nucleosomes. You have this is a picture. This is a textbook's picture. I'm not sure this is correct. It is, but it has some of this. It goes twice around, goes continue, and this is probably folded. So there is more into this picture. But from the analysis at a order of hundreds of milliseconds and more.
Well, I
The
would say
approximation-
you can probably fit the data by adjusting the parameters in the Rouse model to
You can extract
the
information,
effective values of the parameters from.
but however, alpha is something that is more intrinsic. And when you get this alpha, basically you get some information about the statistical properties.
Yeah.
So you have to fix, for example, if you do a simulation,
Yeah.
you say, let's fix this is what it represents, and then you work with this scale, saying that, of course, at lower scale, this is the physics is certainly completely different.
But, okay, my other question is, what about the excluded volume part
Yeah.
of the problem? Because that you can't scale out just.
No, this is much harder. So this is the simplest of the model that account for some of the phenomenology of the data. Now, if you want to get closer and closer to reality, you have to account for Lennard-Jones exclusion. You can also have some bending properties and torsions. Tell me that yet, when you look at this and get the behavior, the asymptotic behavior here, it tells you a lot about with this very rough model about some statistical properties. And what we have observed from the statistics of the data, you still can account for a large fraction of them without putting all of this additional information that you suggested. Again, this is like we have a cutoff at 3 or 4 or 5 kb, kilobase pair. We don't care about what happened below the scale, and then we try to look at these long distances properties. Your question is actually important and very important in the following context. Now let's suppose you do UV damage. I'm okay with the, it works?
No,
No? What shall I do?
what you told me.
Okay. Is it okay? What was it? Yes. So recently, we had the following data where you irradiate a cell with UV light. Now you create damages. And now became, the new player came, which was the relocalization of the nucleosomes. That is, you have this structure, imagine you have a bead here, which is, let's say, hundreds of base pair, of, let's say, 500s. And they start to move. And this is complicated because if we do that slowly, you have the DNA that will wrap, I don't know, 1 or 2.5 or 2.7, a certain number of them, and then it moves to the next one, and then move a certain number of, not a complete circle around. And this guy is starting to be relocalized. Now, this is a very touchy question because this model is not anymore valid. Now we have to account for this localization that they have found experimentally and try to address the question of how the DNA has changed its organization, where now we have this additional player. For example, what happened when 30 or 40% of these guys have moved? How do you model this? You cannot anymore use the classical RAS. How do you model this? So if you go now to, there are models in the literature about looking at the interaction between this nucleosome and the entire chain. It is very complicated. The time scale, if you do a simulation with this, is very short, much shorter than minutes or tens of minutes that you want to achieve with your simulation to look at how things are redistributed. So basically, you are stuck if you do simulations that account for all of this local interaction. So here, I have no answer for this. This is the construction. A sort of hybrid model should be constructed where you look at this nucleosome, how they are going to be redistributed, and let's take this RAS polymer as a backbone of the structure. Now you have to couple the two in a coarse-grained manner so that you can run long-range simulations. So this is we're trying to look at this. This is this is an important point, and the motivation came from this UV light, where you can see beautifully that everything is extruded from this region, and then things start to come back. And then they looked at what is the fraction of nucleosome that went out, and are they going out of this region because of translocation on the DNA, or because they went outside and then they can float like diffusion. So that's okay. So there is no absolute model. A model depends on the questions. Here we're looking at long-range processes and also rare events such as looping. And we will use also this information, alpha, again, coarse-grained statistics that can be extracted from data that can now be related to the interaction in this coarse-grained polymer model. Again, there is a big debate in the literature about what is alpha, where it comes from, what it means. Here we propose that just following the step of the analysis, we give some interpretation. I'm not saying that, you know, it's true, but at least there is something. And we have this procedure of construction. Okay, so let's continue. From here, we have two options. I can show you the mean square displacement. I think I'll start with this. How it is calculated. It's a bit systematic, but at least I'll show you the steps. And the detail of all the calculation are in the papers, but at least I'll explain just the detail. I like calculation because this is my job. Mathematics for me is about how to calculate and how you see things happening in calculations. And then we will move on with the looping, how long it takes to loop and how you do this calculation with these different methods. Okay. And yes, and also there is, maybe this will be for next time, this how to extract Kevin answered the question about the force. How do you extract the force applied somewhere when you look at something at another position? Okay, so let's concentrate on the mean square displacement property of Ross, and so I'll do it in general for beta polymer. So let's continue. So we had the equation of the polymer model, the Ross. From this, we could estimate what is the Holstein-Uhlenbeck process. From this, we can calculate, so the mean is going to be 0. This is classical Holstein-Uhlenbeck. And then from this, we can calculate the mean square displacement. Just this is mean square displacement for Holstein-Uhlenbeck process, which is very well known. So I'll start probably writing here. So what is the mean square displacement for a one-dimensional? And if it's three-dimensional, it's x, y, and z. So you just have to multiply by d, the dimension. Let's assume everything is isotropic. Okay, so sigma p of t, which is the mean square displacement for this Holstein-Uhlenbeck process, is kappa is Boltzmann constant temperature divided by kappa p, 1 minus exponential minus 2t tau p. So this is a mean square displacement. Let's call it for xp or for up. So this is really up t. Let's say times 0. This is 0 squared. No, there's too many squares. Equals. Okay, so let me do it in dimension 1. If you are dimension 3, you have 3 times, assuming that everything is isotropic. And then kappa p is what is written here. And for sigma, this is for p different from 0. And if p equals 0, this is just Brownian motion. So this is sigma t. It's just, this is the motion of the center of mass. So you don't care about the polymer because you look at the motion of everything together, which is like Brownian motion, except that the diffusion coefficient, when you have n beads connected, is going to be d divided by n. So d center of mass, t, and d center of mass should be written somewhere. It's d divided by n. And I need to explain what is tau p. This is 1 divided by d kappa p. So I will write now either kappa p for what you have when you have beta, or when you have 2. Let's think beta equals 2. It's this case. So now let's go back. We can write the expression of rc square at time t for the mean square displacement. So let's take this guy, square it, take the average of a realization. Alpha Cp, this is a matrix which is independent of any time. Up is a solution of Holstein-Uhlenbeck, and we just calculated here the mean square displacement. And since all up here are independent, because obviously, you have one here, it doesn't depend on the other one. So they're all independent. So now when you average and you have the 2 of them, they're going to be equal to 0. So it's equal d. D is the dimension. For example, d equals 3, dimension 3. So this is u0 squared plus the sum, d times, the sum of 1 n minus 1, alpha pc squared multiplied by up squared average. And now we just replace everything here. So this is 2d c center of mass times t. This is just the motion of the center of mass. And now we have to write plus This is better like this. D. So let's replace by kappa P, kappa Boltzmann constant temperature divided by nothing, because the sum, the kappa P is going to be inside the sum, kappa P, and then 1 minus exponential minus 2t divided by tau P. All right, from P equals 1 to n minus 1. Is this correct? Yeah, and now we are missing here this alpha pc squared in the sum. So the sum, there is a sum in alpha p, in kappa p, there is tau p is defined here, kappa p is defined there, and alpha pn is defined over there. All right, so now let's just look. So now we are exactly interested in how this term behaves. So basically, if the time is very, very short, extremely short, I mean, there are 3 regions. What are those 3 regions? Now, just let's look at this.
I'm a little lost. What's the physical interpretation of R? C?
It's a point on one of these points. Any of the. I'll take now a point in the middle. I look. I calculate from this point, according to this, the mean square displacement. At this point, at this moment, we just follow these calculations, mean square displacement of this point, and now we are going to look at what can happen if there are 3 cases. If the time is very, very short, extremely short, time is about 0. Basically, what you're looking at is the position of a bead that hasn't, the time of the interference with the neighboring is not reached. So this is just really a diffusion. We are going to extract information about diffusion of the bead you are looking at. This is one time scale. The second time scale, suppose you wait very long time. Everything has a time to move, and what you are really looking at is the motion of the average motion of the bead, which is really like the motion of the center of mass. In between, this is where we have this anomalous regime, and this is what we would like to calculate, how to express this sum here in this intermediate regime, where you have a time t in between those time characteristic tau p, and tau p are related to kappa p according to this formula, and kappa p is in here. This is called those are the different time scales involved in this relaxation of the polymer. All right, so let's say 1, this is diffusion regime, time very short. Diffusion, so now let's be precise. Time very short, so it should be less than time n minus 1. So this is the diffusion, and we have basically RC squared, which behave like dt. Now, if the time, so this extreme t bigger than tau 1, and basically RC behave like 2d, d is the dimension, d is the center of mass, time t, where dcm is d divided by the number of beads. And in between, so this is not the anomalous regime that we would like to discuss. So when you are in between those times, and so for this, what we need is to look at this sum. So let's rewrite, so let's call it, I call this or just, okay, everything, let's call it Sn. This is the sum here. So now we are interested in estimating Sn when we are in the time scale between tau 1 and tau n minus 1, which are the extreme parts, I mean the extreme time scales. So this is d divided by kn. So k is now just the parameter sum of cosine squared 2c minus 1 multiplied by p pi divided by 2n 1 minus exponential should be minus 2t tau p, and then we have to divide sum for p equals 1 to n minus 1, and now we have to sine to the power beta p pi divided by 2n. All right, so now think about this sum as an integral, where you are going to see that p and time now are going to be mixed together. And so how do you do this? We can use, how to replace this by an integral in a correct manner, I mean in approximation manner. We are going to use the Euler-Maclaurin formula. So let me remind you what it is. So if we have a function, if we have the sum of a function, a regular function, f of p from p equal 1 to n minus 1, this can be written as the integral from 1 to n minus 1 of f when n is large, sufficiently large, f of x dx, plus there is a residual term, f of 1, f of n minus 1. This is found, was found by Euler and other people some time ago, 112 f prime of n. So this is why we need some regularity. You have different derivatives, minus f prime of 1. This is for n, true for any function, so let's use this for that function. Let's plug everything in here. And so let me concentrate for this calculation on the integral part. Of course, we need to calculate the other term to see how they contribute, but just to give you how it works, let's focus on how we calculate this. So this can be, we can split it in two integrals, the integral where you have 1 and the integral where you have the exponential. So this is going to be a sum of two terms, I1 plus I2, where I1 is going to be minus integration from 1 to n minus 1 of cosine squared 2c minus 1 p pi divided by 2n divided by sine of beta p pi divided by 2n, and then I'll take the one with the exponential. So this is why they have the minus tau p. Right, and there is the second term, is the guy where we don't have the exponential. This is a term with, so okay, let me write it. I2 is integration from 1 to n minus 1, basically the same thing. And this is very important here. This is dp. Integrate with this p variable. And now this is the same, cosine squared divided by sine beta, p pi divided by 2n, cos squared 2c minus 1, p pi divided by 2n, dp. Right, so this minus this is the f here that corresponds to that term. If n is very large, then for example, where is the expression? Yeah, so p is fixed. Let's suppose p is fixed, and let's go to the approximation of sine x by x. So we can approximate this by x. The first term here, when n is very large, c is fixed, we are going to make the approximation that it is 1. So I'll make some approximation, not necessarily I'll justify it here, but let's do this. When you do then all this, you can check by looking at the error that what I'm saying is correct. I'll just give you the leading order term. So leading order term, so let's write this. Minus t divided by tau p. So let's go slowly. Tau p is there. So this is divided by, so if I divide, I multiply by d, and kappa p. And kappa p is 4 kappa multiplied by sine. So squared, let's write it p squared pi squared divided by n squared. Is it correct? No, 4. Yeah, should be correct. So this is like 1, and now we have to divide by pi p, so p pi divided by 2n to power beta. And then we have to integrate this from 1 to n minus 1, plus some residual term, and we have to integrate dp. Okay, so now n is very large. We're going to use a change of variable here, and this can be written as the gamma function. So let's go slowly about this calculation. All right, I'll go to the board here. So let's do a change of variable. So in the exponential term, we get, so let's write u equals p to power beta, t divided by tau 1, and tau 1 is 4 kappa d, 4 kappa, yeah, pi divided by 2n to the power beta. Should not be beta here, should be 2. Okay, so now this is exactly the term here. So this term, I1, is going to be dp, so let's write it du, so we'll have to change variable. So we have exponential minus u, this is now u, and we have to replace this term here. And so when we do the calculation, you get u to the power 1 divided by beta minus 2, exponential minus u du. You have t divided by tau 1, n minus 1 here to power beta, t divided by tau 1. And there is a term here, which is t to some power 1 minus 1 divided by beta. And then there are other terms. So this is minus 1 divided by beta, 2n to the power beta, pi to the power beta, and there is also tau 1 to the power 1 minus 1 divided by beta. I skip some steps, but basically you just write this as u. You express p as a function of u. So if you have something to the power beta, basically the p that is going to be here is going to be, so like p squared something is equal to u. So p is like u to some power, p some power u 1/2, and then you have a beta here, and then because of the dp you have another term here. That's why you have this term coming from here. You have the minus 2 coming, and so now you have to study this integral. And so we have to look at what happened in the different regimes. In this regime, this tau 1 goes to 0 because n is very large. So this limit is 0 when n goes to infinity, because if you have 1 over n. This goes to infinity because this is compensated by n to the power beta. This is where you start to get some constraint on beta. N to the power beta divided by tau 1. This has to go to infinity. And this is going to be represented like a gamma function. And then you get here the exponent t to the power 1 minus 1 divided by beta. This is how you get it. And then there is a constant here that, of course, asymptotically depends on some quantities. So you end up having something of the form 1 divided by beta, 2n to the power beta, divided by pi, t to some power tau 1, everything to power 1 minus 1 divided by beta, gamma of 1 over beta minus 1. So the minus here, and this is negative, so this multiplied by this is actually positive. And then what you get is you can estimate the first term. So this is this term here. Now you can estimate the term here. And then you can calculate all the terms. And you end up with a formula. Again, I'm not giving all the steps here. But the calculation is interesting because this is where the t to of this non-integer power comes in the calculation. So RCT or t plus 0 minus RCT 0, you average this, make it squared, and this is of the order. So this is 2d center of mass t plus a constant t to some power 1 divided by beta. And then there are other terms that coming from the other element that I haven't calculated. It's 1 divided by minus t divided by tau 1 for t bigger than tau n minus 1 and less than tau 1. So basically the most important term is this one. The rest you can neglect. And you get this power here with the beta that is prescribed from the alpha, I mean from the exponent, so that if now this is equal to alpha, how we did it in the calculation, we found the alpha, we then use it to fix beta, and now you go to the construction that we've made, and now you can look at your simulations, simulate this, and then you can even extract and make sure that what you have is follow the theory. That there is no corruption due to all of the approximation that we did. You can get this exponent. Now what is interesting is where do you do this? Now you have data. How do you extract an exponent like this? This is a tricky question. Suppose I give you the data. So now, this is a remark. All right, we have some theory with some approximation and some leading order term. Now, if you look at what the data, what it says here, it says that there is some time scale that you do not control, and the time scale here, so it has to be not too small, not too large. But how do you how do you extract the information? Usually you end up with curve, this is the MSD, as a function of time. So if you have something perfect, you get a line here, and usually whatever is your process, you get, and then this is, let's say, confined Brownian motion. You go to here and say, right, I have to approximate the derivative at 0. But you cannot do that here. It has to be long enough. So how do you do? Well, it's not clear. It's not clear. I'll show you what. So we could find that there is an anomalous regime. So at the beginning, it has to be like diffusion. It's very, and back it has to be also very small. But it turned out that there is a regime, an intermediate regime here, where you go. So you go back here to some kind of tangent that has to be parallel to. I mean, has to be go to 0, so you have one like this and another like this. It's not very going to 0, but imagine that it was going to 0. So then we use function of the type t to some power alpha in some intermediate region, where indeed you could see that it was matching because we knew from simulations. But you could see deviation if you don't do it correctly. What is interesting is to have here a certain time window that is sufficient. I have no criteria to tell you where you should start looking at this, and at the point for a process that you don't know what is the time constant. Yeah.
If you rescale with t to the half or something, you'll get one plateau on the left-hand side and a different plateau on the right, and some transition regime in the middle, won't you?
Again, so you want to scale by t to some power, but what is alpha? We don't know alpha.
You know at the left-hand end it's going to be a half, and on the right-hand end it's going to be a half.
That's right.
So if you plot it against that, then you'll get a plateau.
Okay. So you're saying rescale your function f of t by t to 1/2 here, where you have something constant, and you're saying when you see the deviation, put your. It's a good idea.
Yeah. You'll see
We haven't tried that. Yeah, probably this is something to try. Yeah, I have.
You're going to run into enormous trouble with this because the right-hand end is the statistically least controlled end.
You're saying by when you do this, because of the t here, you might have some large fluctuation when you divide by something very
The
small.
fluctuations at the right-hand end are gigantic, right?
That's right.
When you get one piece of information from each track. At the left-hand end, you get one piece of information from each step. So I'm not arguing with you, I'm just making a totally separate point, yeah.
Yeah.
Okay, go ahead. So you're saying that in here, so it's true that here you get this all fluctuations. In here, if you divide by 0, basically, when you do this, it's true that it can also lead to large fluctuation because you are close to the origin. This is what you had in mind?
I'm saying the left-hand end is going to be solid. You're going to be good at the left-hand end.
You mean here?
Yeah.
Yeah, I mean, if we...
The right-hand end is going to be problematic, and so finding the transition, you really, I think like you said, you want to look for the transition from the solid left-hand end to something different, right? Rather than looking from the right-hand end just for a transition there, because you're not going to have any idea about the right-hand end.
Okay.
I think that's what I'm trying to say.
So what we thought about how to analyze this is to say now, because we have some kind of uniform expansion, I mean, we know that it had to start at the beginning with t. Then in the intermediate regime we have this. Suppose now this is uniform, suppose. Why not, instead of just looking at the 3 pieces, just use a formula like this to match everything?
Just fit it.
That's right. Nobody has done this. I mean,
It's a reasonable thing to do.
yeah, this is not that old, you know, we published that maybe 1 year ago. This is something that, this is a type of idea that we had in mind. That go now with the asymptotics of, let's assume this is an asymptotics, and then it is valid uniformly, which might not be necessarily true, but at least now you have, let's say, a constant here, a constant there, you have 3 or 4 constants. You don't want too many to fit a curve, because then you can do just, but if you have some constraint, probably what you said, you know, about the beginning, that we can have in some region some good estimates, as Dan said, probably at the beginning of the diffusion coefficient. Probably in the end, we are going to have a lot of fluctuations. But if we have something uniform, that might be something to try. Look at this uniform. So just, I'm sorry, this is, I should have done something probably on the big screen, but this is what you have. So at the beginning, you don't know. I mean, red and green are the fit to t to some power. This is what Dan said at the end, where you get this fluctuation, it goes to the roof. And at the beginning, you see that there's some deviation because it has to have a different power law. So then there is a region here where you can use this t approximation, and we could extract from this compared to the simulations.
Can I ask some probably stupid question?
Go ahead.
So, tau 1 is just basically 1 over the spring constant, right?
Tau 1 should be, so this is now you look at 4 kappa. So this is p squared, pi squared divided by 4n squared. For p equals 1, you get what it is. So this is pi squared divided by n squared.
Oh, so that's the smallest one. Yeah,
The
okay,
small-
sorry, yeah. So
yeah.
that's the softest mode.
Yeah.
It's the influence from far away, right?
This is the influence of, yeah,
It's the long wavelength, right?
That's right,
It's far
that's right.
away, right? Okay. So, okay. And then your other one is
the one that then you start to have,
now n minus 1 is your neighbors.
that's right.
Is that right?
Yeah, n minus 1, because here you get this n minus 1. Now you get something where, you see,
Yeah, yeah.
this is the n
I'm just
and
trying
it
to figure
goes
out
away.
where around they're going. Yeah.
Yeah.
Right.
Yeah, this is-
Okay, so yeah, this is basically where,
How do I estimate? So that means I should be able to estimate kappa, I guess, from this. A sort of effective kappa.
how do you want to, you mean by,
Or do you know kappa for however long your pieces are, and then you can estimate tau 1? Because how am I going to know my experiment is giving me the, I mean, what frequency do I set the camera in the experiment?
well, okay, here there are interesting points. So to come back to your point about what is the model of your polymer, what should be kappa? Kappa has to do with some mean properties that you define, that you decide. It's related to B, the mean square distance. So at this stage, it was very hard to get information about this time scale. We don't know, I don't know how to do it. And we focused mostly on this anomalous exponent for, because this is what people are interested in. Now you are saying, let's be more precise and define your polymer model according to some data that will fix, for example, kappa. Then you can get some idea of your time constant, which is the time constant that you will get over there, the extreme left and right where you have diffusion and diffusion. This is what you are saying. Get information from your model, from data, and be then more accurate in how you fit. What I can say is this.
Now, what I'm concerned about is you do your experiment and you fit it, and really, because you're sampling very, very slowly
Yeah.
compared to how fast you should be sampling, really you're picking up something completely different than what you think you're picking
What do you mean? What do you have in mind?
up. I don't know.
Here, you should pick up at the time scale the anomalous exponent of your system. This is, what here would you expect to get? What?
So the anomalous exponent is not due to the polymer; it's due to the environment.
How would you have? Okay, how do you make such a statement based on what? That's the alpha. What the environment would do in order to influence the anomalous exponent?
I don't know. I'm sure we can think of something, but you can always find a reason for anomalous behavior. But if you can't sample at the appropriate, there's a time scale here on that graph that you drew, right? And that time scale, I think, is basically set by the stiffness of the bonds, and possibly the number.
The coarse graining. It's bounds. It doesn't. The meaning is what is the meaning here in terms of base pair? We have to be big enough. So okay. So what, for example, we did in some analysis is to change the sampling time to 300 to 80 milliseconds, and ask them, what do you observe? What should be? Is there some significant changes? And there were. So we found, for example, alpha was different, which is not clear. It should be independent. Obviously, this has to do with the problem of how you get alpha, how you estimate alpha. Again, this is new stuff, so it has to be tried and tried and tried to see how you refine. But I think in general, once you have your polymer model, you define a scale. Once you define a scale, using the correlation function, actually, you can get information about the relaxation time, which has to do with this. So we use also this to get some estimate about this time scale. You use the correlation function as a function of time and see how it decays. Okay, I think let's finish with a remark. This is a remark how you connect to data. In the last 10 minutes, let me start the mean first looping time, how you calculate looping. So this is, so I can give the references. This is Amitai. So we wrote a couple of papers on this, but I think one is enough. And myself, this is phys rev E, I mean, there are physical review later, but I mean, all the explanation is really in the Phys Rev E. This is in 2013, Asaf Amitai, where we explained, all of the idea here. You do the simulation. How you do the simulation? You do the extraction and the simulation, not on data. And then we have a paper which is which is submitted, where we apply all of this to real data about double-strand DNA break. Now you have a break. You look at the break before. I mean, the locus where you look at you have tagged your DNA before the break, after the break, and the locus is close to where you have induced the break. And so we can see differences before and differences after. And this is where we see the differences also at the scale at which you are sampling. It's not clear to us, I mean, what it means. Obviously, when you sample at a longer time scale, more of the long range time scale is coming, the modes, the different time scale in kappa p. If you wait sufficiently long enough, more of the polymer will contribute to your dynamics. This is the statement that we have so far. It's not quantified enough here. All right, so now let's go back to looping. I'll do looping, and next time we will do the extraction of the force. So how do you calculate the looping? The looping also has to do with a time. I mean, it's not a time scale in the polymer mode. It is not contained really in this mode here. This is a mode of relaxation of the polymer. Looping is something else. So the looping time, which is really the mean first looping time. So this is the time it takes so we have bid R1, okay, Rn, and you're interested in when this bid is entering for the first time in a ball of radius epsilon of R1. So let's define tau. This is the first time that R epsilon or Rn t minus R1 t for a Ross polymer, this is less than epsilon. Condition that, of course, at time 0, R1 is the distance between R1 and Rn 0 is strictly less than epsilon. It's whatever it is. For example, it is given. You have a given length. So this is, now we're interested in calculating the mean. This is what we're interested in. Mean first looping time. How you calculate this time asymptotically. So R1, Rn are satisfying the Ross equation, which is minus the gradient phi. You take the gradient with respect to Rn plus square root of 2d and the noise. This is for all of them from, except the first and the last, from 2 to n minus, I mean for all of them actually,
David, can I just ask a question?
Yes.
Isn't another question that you're interested in the looping of a subloop in a very long chain?
Yes.
So I mean.
Yeah. So here I'm looking at the two ends. We can also,
I understand that.
you can also do it for the two in. That's right. You can have let's say A and B. And this is what you are anticipating. What is important is not the entire chain. This is the, let's call it N and M. So instead of having capital N in here, you have N minus M, absolute value. This is what you can show. Yes. And this is important. It doesn't matter what happened. Yeah.
Explain
Yeah,
to me again the initial condition that you're using. What's
Yeah,
that?
initial condition, you have to be outside.
But if you're just outside, it won't take very long. If you're
No, but in average, if you average of a steady state distribution, let's suppose here you take time zero, you take a steady state distribution that in your sample of, realization that, the probability they are inside at time zero is small. So let's, for example, assume, you can assume, for example, that time zero, I don't know, Rn is equal to, what really we did here is we took steady state distribution at time zero, assuming that you're at steady state. Steady state is given, you have basically a Gaussian distribution for the position. All right, so this is the phi, this is the potential well that we have. This is R1, Rn, which is kappa divided by 2, the sum of Rk minus 1 squared from 1 to n minus to n. Okay, so now let's give an interpretation of this quantity. This is actually quite nice. We can rewrite this Rn using this. That is, you invert this matrix, and the condition is that it's going to be a condition on UP. So why is it interesting to go into this UP state? Because first of all, we separate U0, which is really translation, which has nothing to do with looping. Translation is just the motion. So this is out. Now suppose you move your camera with your polymer, so remove the translation. Now we are left only with rotation, which is U1, Un minus 1. And so now we are going to give a condition. We are going to work on the space U1, Un minus 1, which is the right space to work on. So now, what is U1? So the space, well, we are looking at the problem. So we take the entire monomers, the entire polymer, the joint, we're going to work on the joint probability distribution function of U1, Un minus 1. So for example, if you start in dimension 3, they're all in dimension 3. So you are, let's say, in the space R to dimension 3, N minus 1. And so you can rewrite this. You invert Rn as a function of UP. This is an orthogonal matrix. And now you rewrite this condition. And this can be rewritten the following way. 2 multiplied by square root of 2 divided by n, sum for p odds of cosine pi p divided by 2n. This has to be less than epsilon. So this, again, you do the calculation, you find that there are cancellation. Now we are back to the Ross model. Ross beta equals to 2. So this is Ross, inverse passage time, for a Ross polymer. So this is the condition. So now let's understand what we are looking at. So now we have the space. You have all of your variable. And basically, you're done when you satisfy this condition, which is exactly rewritten this condition with this transformation. So let's write now the submanifold. This is U1, Un minus 1. Let's call omega tilde, this is omega tilde, is exactly R3 n minus 1. So such that you have this, so S0 is when you take this condition equals 0. So UP cosine P pi divided by 2n. This is exactly for P odd. This is exactly I've burned already 2 pieces of chalk. So, UP equals, what did I say? Yeah, 0. And so now you're interested in S epsilon. This is going to be, so in your space, there is a subdomain, which is really a plan here, because UP is like, let's imagine you have x1, xn. This is hyperplan. And S epsilon is what? Is not the ensemble of points that are at a distance of S. So here there is a square root of 2 that I don't discuss. This comes from the normal, because of the norm of the normal to this hyperplan. So you're interested in what is the mean first passage time. So this is coming to a question where you start. Suppose you start outside, and we will see that there is a Gaussian distribution behind. So you start outside. You have basically, so you have your space. You are removing an ensemble. This is S epsilon. So let's say you start uniformly distributed outside. So we will average over the initial distribution. And so you have a dynamics, which is the dynamics of the U1, UN, which is all our Oshlin-Uhlenbeck. And so you move in your space, and you're interested, when are you going to hit this boundary for the first time? So it's a mean first passage time problem, but you're in very high dimension, R3 to the power n minus 1. Right, so let's write this down. And then let's write, then what is the mean first passage time to this? So now if you're interested, really, in the probability distribution function of the joint, which means U1, U2, UN minus 1 times T. So this is the joint probability of all of the n minus 1 T. So this satisfies, so let's call this P, and I use the index T here. This is P, yeah, all right, PT of X, PT of U. So this satisfies, by definition, the Fokker-Planck equation, which is dp dt equals, so you have Laplacian, so there is, let me put here the diffusion coefficient equals 1, otherwise I have D here. D, so Laplacian P plus divergence of gradient phi multiplied by P. Phi is the potential which is defined here. And now, for example, so the initial condition, you can take whatever you want. It's a uniform distribution. You take P0. P0 of X can be specified later. P0 for the moment is a function. With the condition that absorbing boundary condition, P has to be equal to 0 on this boundary of S. So this is boundary of S. Right? So this is what we have to solve. This is an abstract problem. And now we would like to find how we, so let's from this see where the mean first passage time comes from. And then let's relate the mean first passage time to the eigenvalues of this operator. And then we're going to connect to the math theorem using the, what is the eigenvalue of this operator in the total space when you remove something. This is the connection with the spectral theory. When you remove, in the 70s and 80s, there was a lot of literature in mathematics starting by Mark Katz, where you looked at Brownian motion. So you start to remove an ensemble of sets in a domain, and then you look at when the particle is going to be absorbed. They started with a circle, and then they look at if you remove an ensemble of circles, and of course what is important is the distance between the disk that you are removing. Okay, so let's. So now, what is the solution of this equation, at least formally? This can be expanded in terms of eigenvalues and eigenfunction. So Px of t, if it's just, suppose this is 0, this is just like the Laplace operator. Now here you have to, it's a self-adjoint operator, so you can always remove this, or use the function transformation to have the exponential phi in the eigenfunction. So you have the sum of some coefficient, IA. This is the eigenfunctions, exponential minus lambda IT, exponential minus phi. Again, d diffusion coefficient here is equal to 1 to simplify. And you have to sum over I, so these are the eigenvalues and eigenfunctions. Okay, so what is now the probability, P of t, is the probability of a loop. So this is the probability of tau epsilon less than t, that is, now let's think. If tau epsilon is less than t, it means that you have formed a loop. At time t, you have already formed the loop. Here, at time t, you haven't formed the loop. Is this correct? Yeah, I don't know, it's not written. So let's think. This is a probability that you're still in the domain at time t. So this is integration over omega epsilon of the probability distribution function px of t dx. So omega epsilon is your space minus omega epsilon is omega tilde minus s epsilon. So if you integrate the probability distribution function over your space, you're still inside, so you haven't formed the loop. So this is correct. And so now the probability, so this, if you can take the solution here, you can integrate this from 0 to infinity. You take this over omega epsilon, and you get here a sum of whatever coefficient. This is not going to be a constant. This comes into here, and you get a sum of exponential ck exponential lambda kt. And so now, let's go back to the question. So we took for p of 0, exponential. So this is the distribution of the initial point that you asked. So this is exponential phi over omega. The volume here is the volume of the space with this weight function, exponential phi. So omega is integration of omega tilde exponential minus phi of x dx.
Hold on, don't you need to exclude the points that are inside the...
Yes, all right, this is true. But they are of size epsilon, basically. Yeah, okay. So let's call it omega epsilon here.
Sounds good.
Yeah? Okay. So now the mean first passage time, tau epsilon, is the sum of the integral of this. So this is Ca divided, so diffusion constant is equal to 1 lambda I epsilon. Well, lambda I, so let's call it lambda I for I equals 1 to infinity. Okay, so now, in principle, if you want to calculate mean first passage time, you need to know the spectrum, which is very demanding. So, when we started to look at this using simulations, we could find that this probability distribution function of a loop is basically, you can take something of this form. At least you start with the two terms, lambda 1, lambda 2. This is normalized to 1. So this actually carry about 0.9 when n was, let's say n equals, you do simulations, you have, let's say, at least 30 to 64 beads. And this can carry some of it, but basically, the idea is that the process was almost Poissonian, because it takes a long time. It's a rare event, and basically, you don't care about the rest. Most of it is accounted by the first term, suggesting that the mean first passage time, when you calculate this, can be just given by the first eigenvalue. Now, okay, so I'll continue next week. Sorry, I didn't meant to take 10 more minutes. So I'll stop here, and the next week I want to go to this. So as Dan said, here we have excluded a region. So this is what? This is the eigenvalue of an operator where we have excluded a region. Now, how do you connect the eigenvalue of let's say, the operator where you have no, you haven't removed anything with this eigenvalue? So this has been done, and again, it follows the idea of Mark-Katz. There is a shift, and the shift has been calculated. This is called the Chauvel and Feldman theory. I just write in here so that you may have a test to come in 2 weeks, not next week, in 2 weeks. The idea is that lambda I, even for the entire spectrum, this is for the entire spectrum, is a shift compared to the, so let me, this is the spectrum eigenvalue of the operator which has no perturbation. This is a perturbed one. C2 epsilon integration over S eigenfunction squared for the associated eigenvalue here, integrated over S. So this is really the volume induced on your submanifold. So I don't know how to write this, but this is the dV on S.
So S is the part you removed?
Yeah, S is S0. Yes. So this is a normalized eigenfunction. So you see that it's of this order epsilon, and what is important here is where this epsilon comes from. So somebody asked at the beginning, yeah, I think it was Peter. If in dimension 3 it's 1 over epsilon, dimension 4 is 1 over epsilon squared, why we still get epsilon in high dimension? Here, what is important is that what you are removing here, this, how many equations do we have here? If we are in dimension 3, if you use dimension 3, you have 3 equations. So you have 3 constraints. So if you are in n dimension, basically, you are removing something of codimension 3. And this formula is exactly in this case. If you have only 2 constraints, if you are in dimension 2, instead of epsilon here, you get 1 divided by log of 1 over epsilon. Yeah. So this is where it comes from. In this formula, you are using the fact that it's codimension 3. And so it's epsilon, and this is epsilon. So as we're going to see next week, for the first, if you have an eigenvalue, the first eigenvalue of the Laplacian, if you have a boundary, periodic boundary condition, this is constant. So the eigenfunction is constant, and the eigenvalue is 0. So this is exactly 0. And what we need to calculate here is this integral over the path of the closed loop. And this is normalized, and normalized is the space basically of all the loops. This is what we need to calculate next week. The integral of this quantity, so it's going to be a constant, of this is dV associated to the exponential phi, so this is going to be Gaussian integrals. Because here this is, again, the Laplacian, it's not exactly the Laplacian here because we have the exponential phi. So the first function is exponential phi. Exponential phi, this is the Gaussian. So this Gaussian integral, basically we have to integrate exponential phi over the entire space of all the polymer, and then the exponential phi over all the loops. And the rest of the geometry is taken care by epsilon, and C2 is, I will write down, this is associated to the gamma function. I mean, it's a given value, square root of pi. So this is the structure of this, which is going to be exactly the first order term in the mean first passage time. That's the idea here. And in 2 weeks, we'll go step by step to calculate this term.
Right, Dave, can you just clarify that integral there? That's over the excluded part? Or that's
This
over
is now an integral over of the skeleton. There is no more epsilon here. So you have exactly the loop. It comes exactly, you have whatever it...
The boundary of the loop.
It's exactly the loop. You come back to epsilon equals 0. So you integrate over... Well, okay, let's write it. It's exponential minus phi, so you have to sum of ui squared, something like this, from y to n minus 1. So this is going to be the integral of du1 du n minus 1, but there is a constraint now, and the constraint is that the un minus 1, or depending on if it's odd or not, has to satisfy this. So you can express one of them in terms of the others, put that into integral, do the calculation of the Gaussian integral, and you get a formula. Next week we'll do this. This is you integrate over something of minus dimension. I mean, you have removed, actually, you have removed here, this one is going to be connected to the other one, so it will disappear. And so if you're in dimension 3, the uk or un has 3 coordinates. So you're removing 3 dimensions because you have 3 constraints here. So you plug back into that here, you express un minus 1 in terms of u1, un minus 2, and this is what you have to calculate. We'll do that next week. Sorry, I have been taking 15 minutes more, but I don't have my children today to pick up. That's why I didn't worry much about the time compared to the previous days. Thanks.